Now I'm going to show you the key parts of how to manipulate DNA nucleotides in the mitosis lesson. So the idea here is not to focus on the cell membrane or the nucleolus or any of the other those exciting parts. The centrioles, I mean, those are all key parts. But I really want to focus on the manipulation of this DNA and this DNA structure. So to set the whole thing up, there's a number of kind of structural things I'd like to talk to you about. So in terms of the design of this particular lesson, you can see here we have 4 nucleotides and 4 nucleotides. So the idea is the length of this DNA strand is the same length as this DNA strand. So we communicate to the students that these are homologous chromosomes because of the same length. We're showing them that they are different because obviously we have A and T and G and C. That's even going to become more significant in our next lesson when we do meiosis. The other thing that's important is the positioning of where I've placed the homologous chromosomes. You can see these yellow magnets here. So this, when I set this up, this yellow magnet told me to put this strand here. And when I open this to replicate it, I'm going to move this strand to this position where that yellow magnet is. I need that much distance between the two. And the same thing is true for the homologous partner. This yellow magnet is telling me where to position the T's. You're going to see me open the A's this far. And that gives me the correct position to attach centromeres. You can see I've got the T's set up here to do my replication, and my A's and my C's and my G's. The other thing that's important, and I just kind of came up with this about a year ago. It's a simple thing. I put the red magnets here to communicate to the students, and they would just take a marker and put a dot where my red magnets are, and that would communicate to them that these are the what I call the original strands before DNA replication or synthesis starts. This particular manipulation I'm going to show you, I would show it to my grade 9s, and the same identical manipulation to my grade 11s. I would go into a little bit more detail with my grade 11s about a number of things, but in terms of manipulations, the grade 9s and 11s see the same thing. So I'm just going to remove the mitosis sign, and we can see here our centromeres, worker protein 1, worker protein 2, worker protein 3 and 4, they're all in your legend. To identify these proteins at the grade 9 and the grade 11 level just by worker proteins seems to have been a really effective way to communicate to the students that, okay, these are obviously enzymes, but they're doing work, and that resonates with the students, right? Sir, what is a worker protein 2 going to do next, right? So that's why I'm using that language at that level. So we're at synthesis, at interphase. This is also a very good time to do a before, during, and after diagram. So I'd have my students sketch this chromosome set of DNA and this section of DNA. And what they see right here, that's a before diagram. The during diagram, when worker protein 1 comes and starts to open, we kind of just do a little mini bubble. I can bring in with worker protein 2, 1, open that a little bit more, 1 complementary base pair, and then get another 1. And even right there, I might stop and tell the students, you know, now they use an arrow here. So they've actually sketched this. This is open, an arrow here, and then maybe another arrow here, and that's all they really need for a during diagram. And then, so what I would continue to do here is, if I just back up for a minute, I'm going to move this over to where the yellow magnet is. And now I'm going to do this complementary base pairing rather fast for you. And so worker protein 1 and worker protein 2 opened and did complementary base pairing. Now, worker protein 3 at the same time is creating, in this lesson, a new centromere. So I place a centromere on the new strand, and I'll take this centromere on the new strand, and you can see how I was off here a little bit, and again, it's not a big deal. You can just slide them together so that the centromeres are touching, and that's what the positioning that I'm trying to get. So again, it doesn't have to be perfect, and you can talk about the significance of the centromere. Now, come over here, and same thing, right? So I'm going to open this strand. So that's what worker protein 1 does. Worker protein 2 does complementary base pairing. And again, obviously I'm going fast because all you people are super smart. And the pacing, when you teach this, you'll be watching your students take notes, and it'll be clear to you what kind of pace to use. So now worker protein 2 is going to bond the nucleotides together, and we have now created 2 new strands of DNA. Worker protein 3 is also producing a new centromere. So these are identical pieces of information that are bonded together by their protein centromeres. It's kind of like a poem I wrote, actually. Now, worker protein 4 is then going to come along and simulate moving into metaphase by moving. And see what I'm going to do here? I'm going to put one hand on these 4 nucleotides, and I got the centromere, these 4. So I'm just going to move this up in one move, and up, and up. So worker protein 4 is positioning these chromosomes. Worker protein 4 comes over here. And again, I should be able to do this in one move. And worker protein 4 comes over, and how about I hold on to that puppy while I do this? So now this is the simulation here of metaphase. Now the other thing that's kind of interesting is to teach the students a little bit about the versatility of proteins. We can come back to worker protein 1 and say, look, it can do more than one job, right? So worker protein 1 is now going to break the bonds between the centromeres. So as you can tell, we're at a critical stage of anaphase. So worker protein 1 breaks these bonds. Now they are no longer attached. And like the high school students we're teaching, they're free to move. So worker protein 2 can do a second job by moving this structure in this direction. And the same thing here. And worker protein 2 is going to move this structure over here like this. And one more time, like so. So now we have separated, and you all know that we're going to create 2 new cells. Now, one thing I'd like to show you just before we get to the end of the lesson is another interesting thing. If you just take some of the proteins and begin to give the students a little bit of an understanding of the mechanism, and you can just play around with this, right? But a little bit of the mechanism of, if we have, and you can just use a simple mini version here. So now we can say, okay, what was the mechanism of the movement that I just modeled with my hands? Well, the protein now can become the spindle fiber. And as we know, when the spindle fiber is attached, what I try to get the kids to understand is when this protein starts to contract, right? It's attached to the DNA, and it's going to rotate with it, and that's the movement, that's the condensing of the spindle fiber. So when we show our students that move, that kind of really connects with them, and they start to understand some kind of mechanism of the movement that they've just witnessed. So those are the key components to modeling mitosis. Again, enjoy yourselves.